Next, we'll be hearing from Al-khayyat from Laurentian University in Canada. She'll be presenting her project titled Investigating the Cellular Effects of FRA1 in the CGL1 Radiation-Induced Tumorigenesis Cell Model. Thank you.
So should I start sharing my screen or?
Yes, please.
Sure. Can everyone see the first slide?
Yes, we can see it and we can hear you.
Perfect. So I can begin?
Yes, please go ahead.
Sure, thank you. So good afternoon, everyone, and thank you for attending today's session. My name is Al-khayyat, and I'm a PhD student in the Biomolecular Sciences program at Laurentian University and Northern Ontario School of Medicine. Today, I'll present to you my thesis project that is under the supervision of Dr. Christopher Tomei and Dr. Suji Thermalingam. So when it comes to cancer and how things can go wrong in the cell cycle, there are about 3 main important types of genes to consider. We have proto-oncogenes, tumor suppressor genes, and other types of genes such as DNA repair genes. Now, tumor suppressor genes are genes that normally slow down cell division, repair DNA, or cause cells to die through apoptosis, necrosis, or autophagy. However, when tumor suppressor genes are damaged, deleted, or epigenetically silenced, cells can grow out of control. Now, a tumor suppressor gene is like a brake pedal on the car. It normally keeps the cell from dividing too quickly, just as the brake keeps the car from going too fast. When something goes wrong with these genes, such as a mutation, cell division can get out of control. Now, an important difference between oncogenes and tumor suppressor genes is that oncogenes result from activation or turning on proto-oncogenes. However, tumor suppressor genes can cause cancer if they are inactivated or turned off. Now, the first insight into cellular activity of tumor suppressor genes came from somatic cell hybridization experiments in the late 1960s. The fusion of normal cells with tumor cells yielded cells containing chromosomes from both parent cells. Now, in most cases, the new hybrid was not capable of forming tumors in animals, and thus it appeared to researchers that genes derived from the normal parent cell acted to inhibit or to suppress tumor growth, formation, or development. Now, somatic cell hybrids generated by fusing together 2 different cells from the same species have been extremely useful in the study of genetic analysis of their malignant potential. Now, fusion between tumorigenic cells, such as HeLa, and normal cells, such as human diploid fibroblasts, results in the CGL1 hybrid that is genetically stable, non-tumorigenic. Now, irradiation of the CGL1 segregates 2 new cell lines. One is a non-tumorigenic cell line called CON, and one is a tumorigenic cell line called GIM. Now, our lab has performed a transcriptome analysis between CGL1, CON, and GIMs, and they have found 2 important discoveries. The first one is that there are no differentially expressed genes between CGL1, so the non-tumorigenic cell line, and also CON, which is also non-tumorigenic cell line. However, they have found that there's about 1,000 and so differentially expressed genes between GIMS, which is a tumor cell line, and the non-tumorigenic cell line CON, in which 600 and so were downregulated genes. Further research has shown that these downregulated genes were specifically located on chromosome 11. And further research with GIMS, they have found that GIMS lack chromosome 11. And then if they insert chromosome 11 into GIMS, that will reduce their tumorigenicity in vivo study. And then they have found that if they remove chromosome 11 from the non-tumorigenic cell line, so that's CON, that will make the cell line more sensitive to radiation and eventually will turn into a cancer cell line. So the table here shows the genes that were downregulated, and they are mostly located on chromosome 11. And our lab was further interested to investigate the cellular functions of the Phosphatase gene, which codes for the FRA1 protein. So the summary of that paper is that loss of Phosphatase gene or methylation of the Phosphatase gene promoter leads to a reduction of the FRA1 expression. And then the FRA1 expression is lost in GIMS, so in the cancer cell line. And then FRA1 overexpression, so if they overexpress the FRA1 protein in the cancer cell line GIMS, that will suppress its tumor growth in vivo study. So the Western blot analysis here shows or compares the FRA1 expression, the FRA1 protein expression in the non-tumorigenic cell line, so that's CON. So we have here two control cell lines. HeLa is a tumor cell line, and then CGL1 is a pre-cancer cell line. And then CON is a non-tumorigenic cell line. We can see that based on the Western analysis that the FRA expression is somehow highly elevated in these cell lines. If we compare that to the GIMS, so to the cancer cell line, we see that the different strands of the GIMS cell line actually lack these FRA1 expression. So what they did next is they have inserted, as I said, or overexpressed the FRA1 expression in GIMS, and then that dramatically decreased any tumor growth in vivo study. So the reason we focus on the Fossil1 gene is because it codes for a FRA1 protein that is a subunit of one type of transcription factor that is normally misregulated in carcinogenesis, which is a resident nucleotranscription factor called activator protein 1 complex. Now, since its discovery, the activator 1 complex activation has been linked to neoplastic transformation, and IP1 is a transcription factor that regulates a variety of cellular processes, including proliferation, differentiation, and apoptosis. Now, the transcription factor AP1 is a dimer consisting of either junjun homodimers or jun phosphatidimers. So really, the main objective of my first chapter of my thesis is to determine if the Fossil1 gene identified on chromosome 11 is in fact a tumor suppressor gene using CRISPR-Cas9-mediated gene knockout and activation in the CGL1 cell line. So we have two hypotheses. Our first one is that if we delete the Fossil1 on chromosome 11, that will promote transformation frequency of the CGL1 cell line once exposed to high doses of radiation. And then if we overexpress the Fossil1 on chromosome 11, that will decrease the transformation frequency once exposed to high doses of radiation. So as I mentioned, we are using CRISPR gene knockout and CRISPR gene activation. So what we did is we have created in our lab lentivirus particles that either carry Cas9 or dCas9 DNA plasmid, and then we have infected our CGL1 cell lines with that. We have validated our cell line using immunohistochemistry, so we're confident now our cells actually express the Cas9 or the dCas9 protein component of the CRISPR. Then what we did is we have designed certain RNA components or sequences that are complementary to the Fossil1 gene, and then we have created lentivirus particles, specifically gRNA lentivirus particles that carry an RNA sequence complementary to our gene of interest. We have infected the CGL1 cell line with either the Cas or the dCas9 component. And then finally, we have used Western blotting to verify that these cell lines actually either express an overexpression of the FRA or a knockdown. So very quickly on this, on your left side of the slide, we have two control cell lines. So we have the wild type of the CGL1, and then we have the wild type of the dCas9, and then we have three different versions of the Fossil1, just in case if one of them doesn't work, we have a backup plan. So in here, we found that activation number 3 showed about twofold increase within the protein of the FRA1, which is great. However, with the knockout system, we have also three different versions of the knockout with respect to the Fossil1 gene. And then we found that knockout number 1, based on the stat, gave us somehow a reduction of the protein. So once we have our cell lines verified, we moved into experiment work or assays. So the usual experiment in radiation biology lab is survival assay, gene expression, alkaline phosphatase, and transformation assay. So for the survival assay, we have selected our doses from low doses to high doses in addition to our sham or control. We have seeded certain densities of cells per each flask or per each condition. Then 8 hours post seeding the cells, we have irradiated the cells with respect of each of the doses. And then we have 9 days after that, we have stained the cells to count their colonies to find any survival of the cells. And then what we see within the results here, within the graph, on the x-axis, we have the doses that we have used, and on the y-axis, we have the different cell densities that they were surviving at. So we have the 5 different cell lines, so CGL1 control, CGL1 dCas9 control, CGL1 Cas9 control, and our Fossil1 manipulated systems. So what we see is that as the dose increase, we see there is a reduction within the survival of the cells. We see a slight reduction in survival when it comes to gene knockout with respect to the Fossil1, but that's not significant that we can rely on. The other assay that we have looked at is alkaline phosphatase assay, and the reason why we investigated alkaline phosphatase is because this enzyme or this protein is a cellular biomarker in cancer, so it's normally highly elevated in HeLa or in any other cancer types. So what we have done using the assay, we found that when we overexpress FRA, or the phosphatase gene in CGL1, it does show us somehow a reduction of the alkaline phosphatase assay. And then if we delete the gene completely from the CGL1 cell line, we see there is, a high level expression of the alkaline phosphatase. However, when we run the stats on these numbers, we found that it's actually not very statistically significant that we can rely on. So we have two new other assays that we are currently trying to proceed with, or to move forward with, is the gene expression assay and also the transformation assay. However, due to the pandemic, we were forced to shut down our lab for about 2 months, and now we were planning to start these new assays next month. So very briefly, with the transformation assay, what we'll be doing is we're going to be seeding the cells within specific densities. Then we will be irradiating those cells, and then we will see whether the radiation will force these manipulated CGL1 cell lines with respect to the phosphatase to transform into a cancer cell line or no. So certain key points that I would like to leave the audience with is that GIMs lack chromosome 11, and thus they do not express FRA1 protein. And then if we insert FRA1 protein in GIMs, that will suppress their tumor growth in vivo study. We're currently investigating the phosphatase cellular function in the CGL1 cell line, which is a hybrid cell line, so it has multiple different chromosomes in it. Alkaline phosphatase expression was not significantly affected with FRA overexpression or knockout. And then we're currently working on the transformation assay and the gene expression assay. Now, in terms of people that I would like to thank Dr. Doug Burham, our head within the radiobiology group at NOSIM. I would like to thank my supervisors, my amazing supervisors, Dr. Christopher Tomei and Dr. Sujanta Thermalingam. And I would like to also thank my committee meeting, Dr. T.C. Tan and Dr. Cowboy. And of course, I would like to thank the Beyond Science Initiative Conference for this amazing opportunity. And thank you so much for listening, and I'll be more than happy to take any questions if you have any. Thank you.
Thank you so much. That presentation was fantastic. We do have some questions for you. So what is the function of FRA1 in healthy cells? Yeah, so
great question. It has a variety of cellular function. It can either help in proliferation, differentiation, apoptosis. So if that protein is recognized that there's something wrong with the cell and it's a healthy cell, it will initiate the cells to undergo either apoptosis, necrosis, depending on the pathway. That is within healthy cells. Now, within cancer cells, we found that the phosphatase expression is actually function as either a chemotherapy resistance or radiation resistance, depending on the cell type. However, with our CGL1 cell line, we found that it functions as a tumor suppressor gene, which is quite interesting and the opposite of what's currently being found in the research. So we were interested to see what exactly controls the function of the phosphatase with respect to the different cell lines.
So changing the expression in normal cells will have an effect then. And then why will this be a problem during radiation therapy?
Okay, that's a great question. So because we don't know how a cancer cell really functions, we think that the phosphatase expression is somehow a resistance to radiation, but we don't know yet if that is the case. Within our cell line, we found that it doesn't really, within the CGL1 cell line specifically, we found that it's not really a radiation resistance, but we found that in a different project that in, I think, breast cancer cells, it actually functions as a radiation resistance gene. So in conclusion, if I want to sum this up, we found that the phosphatase expression is not a radiation resistance in a hybrid cell line, so in the CGL1 cell line, but it's actually a radiation resistance in other cell lines, specifically cancer cell lines.
That's fantastic. Thank you so much.
Thanks.